think are really fascinating. And that's the microbes which act to cycle iron in the environment. And I want to tell you a little bit about some work that we've been doing looking at how these microbes could influence mineral-associated organic matter. And since I'm in the Northern Hemisphere, I'm not going to talk about forests. I'm going to talk about Northern Hemisphere peatlands, particularly in the Arctic, as a kind of case study. So I think iron's really awesome, and I hope I can convince all of you that that's also the case. But why actually do I think it's so special? So apart from the fact it's just really abundant in the environment, so be the fourth most abundant element after oxygen, silicon, and aluminium. But the way that it behaves in the environment gives it some really unique properties. So on the face of it, it looks kind of boring because it only has two redox states, iron 2 and iron 3. So you would think that doesn't give it a lot of opportunity to do a lot of kind of transformations in the environment. But the really cool thing about iron is that its redox state is directly tied to its solubility. So at neutral pH, when you have easily soluble iron, you have iron 2, it's dissolved. And when you move into an oxic regime, you precipitate minerals because the solubility of iron 3 is much less than that of iron 2. So in the environment, when we have changes in redox state, which happen in probably any environment you can think of, soils, sediments, lakes, it's a really dynamic system. And these redox transformations of iron can really have a huge influence on these dynamic systems. And one of the ways that that happens is because iron minerals are so good at trapping other things. So particularly when it comes to organic matter, it's really good at sort of trapping and stabilizing organic matter that's in the environment. So that can happen as a kind of co-precipitation, where you have sort of aggregations of organic matter that can be even glued together by these poorly crystalline iron oxyhydroxides. But we can also have absorption of organic matter onto mineral surfaces, and that happens on poorly crystalline and crystalline iron minerals. But then even in the aqueous phase, we can have our aqueous complexes of iron and organic matter. So there's a whole huge host of ways that these iron minerals interact with organic matter in the environment. And we heard about that in quite a few talks actually over the past few weeks, that especially in very mineral soils, the mineral content and the reactive iron content is very strongly correlated with the soil organic carbon content. So we have this link between how much mineral you have in the soil and how much carbon you can kind of stabilize and hold. And I'm going to talk about carbon in this talk, but it's worth bearing in mind that this is also true for nutrients and contaminants. So iron is great at trapping phosphorus, things like arsenic, cadmium, any real heavy metal you can think of will be really readily trapped by the formation of iron minerals. So it links a lot of different things in the environment and can have an impact on a lot of different environmental processes. The flip side of that is that when you reduce the iron again, you release all of these things that were previously stuck to it. So if your organic carbon was trapped, if your phosphorus was trapped, when you change the redox state, when you go into a new regime, then you dissolve all of the things, or you release all of the things into the aqueous phase that were previously trapped. And this iron reduction in the environment is very often driven by microbial processes. So we have microbes that oxidize organic carbon, sometimes hydrogen, or even is more starting to become a bit more understood, even methane. And when they do that, they dump their electrons onto iron 3 minerals and they reduce them. So that's yet another way that the carbon cycle is tightly linked to the processes of iron transformations in the environment. That obviously can then switch over again, and then we can oxidize all of this iron 2 again and form these co-precipitates. And I'm not going to talk so much in this talk about the oxidative side of the iron cycle, but I thought it would be nice to add in a few of the different ways that microbes can act to oxidize iron, because I think outside of maybe the kind of nerdy iron circles that I move in, I think the oxidative side of the iron cycle gets a little bit less press, and it's really, really fascinating stuff. So in the oxidative side of the iron cycle, you can have essentially three processes or three different types of microbes that can oxidize iron. So there's photosynthetic bacteria that can do iron oxidation. And these don't do this in a respiratory process. They actually oxidize iron to get electrons for biomass formation. So they aren't directly taking energy from this reaction, but they use it in order that they can actually just build cells. And if there's anyone who knows much about early ocean biogeochemistry, it's thought that these microbes would have been really important at the very beginning of life on Earth, because there was so much iron in the ocean, you know, a lot of electron acceptors, there's a huge dearth of oxygen. So that you would have a lot of these photosynthetic iron oxidizing bacteria early in Earth history. And probably the most well understood or kind of most famous of the iron oxidizing metabolisms are those that use oxygen. So they use aerobic respiration and use oxygen as an electron acceptor, and that can happen at low pH or also at neutral pH. And then, as actually Francisco has talked about quite a bit in his talk, we can also have iron oxidation linked to denitrification, so to the reduction of nitrogen species. And when Francisco was talking about his system, that's mostly an abiotic reaction. But in a lot of the systems that we look at and a lot of the microbes that we study, they have the enzymatic capability to directly couple the iron cycle and the nitrogen cycle within their metabolism. So I hope I've managed to convince you that iron is really a central part of most major biogeochemical cycles. It's sometimes not so much thought as one of the major cycles, you know, not as important as carbon or nitrogen or phosphorus, but the transformations of iron and the microbes that can transform iron between its different states essentially put iron at the center of a lot of things that go on with these other biogeochemical cycles. So when the iron changes, everything else changes with it as well, which I think is really cool. So the things that we know about microbes that use iron and transform iron and their role in the environment mostly come from very mineral-rich environments, so mineral soils and sediments. And the forest community have done a huge amount in understanding iron, organic matter, correlations in soils. And I put this picture of a tree in because I realized it was a forest thing and I thought I should put in some trees. So this is the only tree you can expect to find in the talk. I didn't even have my own one. So what we wanted to do, and the kind of questions that I want to talk a bit about in the rest of this talk, was how we've been trying to move from thinking more about these microbial iron cycling processes in mineral-dominated environments to moving into very, very organic-rich environments. And the first thing we wanted to know is, could we expect that you see the kind of mineral stabilization that we see in mineral soils also in very organic rich soils? And what is the role of microbes in the stability of these iron, carbon, iron organic matter associations in very organic rich soils? And this isn't just a sort of academic exercise where we thought, okay, that would be an interesting question. It's actually got a very important kind of environmental reason for looking into these questions. And that's because actually, if we're interested in carbon cycling and the future of carbon cycling on the planet, we have to also understand the places where carbon is concentrated. And that's actually about a third of the carbon on Earth is concentrated in peatlands in the Northern Hemisphere. And that's mostly in zones which have permanently frozen soils or permafrost zones. Because it's cold, we have low rates of degradation, and we don't accumulate, we can accumulate organic matter more effectively in these regions. But I'm sure everyone on this webinar at least is agreed that the climate is changing really dramatically, and especially in the Arctic, where the Arctic is warming faster than anywhere else on the planet. And it's expected that a lot of these permafrost regions are warming, and that we have a huge amount of thaw expected over the next few years. And this is quite an old map, but I find it really simple and quite intuitive. And it shows the regions in red where we can expect to have completely lost permafrost by 2050, and the areas in a darker orange where we expect them to be gone by 2100. So that's a huge amount of permafrost loss just over the next 80 years. And that has big knock-on effects on how we think about the way that the carbon cycle might evolve over the coming decades. Because if you melt all of this permafrost, you expose a lot of organic matter for degradation, and you also change the hydrological conditions into a totally different regime. And changing hydrological conditions means changing redox states. So that's why I find this question so interesting, because I really like to know what happens when you have these big shifts in redox regimes. And of course, that leads to an obvious question of what impact might that have on the release of carbon into the atmosphere and any feedback mechanisms that we might expect. So I'm going to tell you about a study that we've been doing in Sweden, where we had a couple of simple questions. We wanted to know whether iron was capable of preserving carbon in a model peatland, and what happens to that iron-associated carbon pool when we have permafrost thaw. And I should say before I start anything that this is all work by Monique Patzner. So she's one of our PhD students in Tübingen, where I was before I moved to Bristol. And she's really pioneered all of this, and this is all of her work. So she definitely needs to get the credit for that. So where we've been working is a place called Stordal in Meyer. It's, I'd say, quite famous in kind of permafrost circles. It's very well studied and well understood. And it's in the north of Sweden, just here where Norway kind of comes over the top of Sweden. And it's in a really vulnerable permafrost zone. So this is a place where we expect that there won't be any permafrost anymore by about 2050. The permafrost here is already what we call discontinuous. So we don't have huge areas of intact permafrost. It's very patchy, and it's mostly just small kind of bubbles that are really left. And this is a drone image of our peatland on a really, really lovely day. You don't always get nice days like this when you work in the Arctic, but this was really quite a nice day. And the thing that is so nice about working at this site in Abisko is that we can study different thaw regimes across a really small space, because we have active degradation and an active loss of the permafrost. So we can split our site into three kind of broad definitions. We have these Palsa regions. They are underlain by intact permafrost. So they're kind of raised up about 50, 60 centimeters above the ice surface. And they're generally quite dry, they're quite oxic. They have a huge amount of organic matter and mosses that have built up. But when the ice melts, this Palsa collapses down, the ground surface is lowered, and we move into this more boggy regime. So in this scenario, we still have some very deep permafrost, but the active layer is much thicker, definitely a few meters at least, and it becomes very wet and really, you know, the kind of place that your wellies are falling into the mud. It's a totally reducing, very wet regime. This is fed by rainwater, and by water that runs in off the palsa. That means it's quite acidic. So it's about pH 4. But when this bog region kind of gives way to the south of the mire, we move into a regime which is more what we would classify as a fen. So this means that it's not only fed by rainwater, but there's also a groundwater influence. And the main difference that this has is that we have a dramatic pH change as well from the bog into the fen. And we expect by 2050, at probably the latest, that all of the palsa regions in this area will have collapsed. And you can see how much must have already collapsed over the last few years, because if you look at our little igloo, which is here, I think if it was to be decided nowadays where to position the igloo, it wouldn't be so precariously positioned at the edge of the palsa. So when that was put up, it was obviously on a very stable surface, and that's slowly collapsing all the time. So the work I want to describe to you now comes from when we went in 2018, Monique and we actually got stuck in something called the Scandinavian heat dome. So we were doing field work in the Arctic at about 35 degrees, which was a bit weird. But what we were doing was taking a bunch of cores along this transect from the palsa into the bog and into the fen. And we were interested in, first of all, collecting pore water. So we did this back in the lab, in the glove box, using rhizome samplers. So we tried to keep everything anoxic while we're processing the samples. And then we split the cores into different horizons. So we deliberately do this because we have some compaction during coring. So we don't really trust our depth intervals so much. So we take those with a little bit of a pinch of salt. So we tend to work on horizons. In a really special scientific site like this, digging a soil pit is completely out of the question. So we have to sort of live with our compaction issues for the foreseeable future. So I'm going to do a bit of a run through quite a lot of analysis types that we did. So one of the main things we've been doing are what we call selective dissolutions. And those are really familiar to a lot of the soil scientists. So where we try and separate different crystallinities of iron into different fractions. So we look at total extractable iron with a 6 molar HCl extraction. We look at reducible oxides using dithionite citrate and hydroxylamine HCl. And we look at the colloidal or organic matter associated fractions. So it's a very soft, poorly crystalline fraction with sodium pyrophosphate. So these different extractions just extract different crystallinities of minerals. We also look at the pore water geochemistry, so things like iron redox state, DOC, fatty acids. We've done a bit of X-ray absorption spectroscopy to look at the mineralogy, a little bit of nanosims, and we've also been cultivating some iron-reducing bacteria to estimate their abundance. So I want to talk to you a bit through our selective dissolution data. If you look at the palsa on the left-hand side, you can see in the orange and the hashed lines that these are our reducible iron fraction. Oh no, my light's gone off. I'm going to have to turn it on. Sorry, my lights go off if I don't move enough. It's a bit weird. Okay, so if you look at the orange and the hashed lines, those are our reducible iron fractions. And if we look at the palsa, we can see that they're about the same size as our 6 molar HCl extractions. That's our total iron. So in the transition zone in grey and in the deeper mineral horizons in yellow, we have quite a lot of these reducible iron. And what we find as we go along the thaw gradient is that the proportion of reducible iron gets less and less of a percentage of the total as we move along towards the fen. And our mineralogy results supported that observation really nicely. So when we look at the EXAFs, we can see that the relative amount of iron-3 organic matter and iron-3 oxides in this transition zone of the PALSA is really high, so it's almost 90%. And even in the mineral horizons of those PALSA soils, we still have a lot of reducible oxides and iron-3 organic matter. But as we move towards the fen, this really gives way to clays and iron sulfides, which are a lot more crystalline, and we lose this iron oxide fraction. And what I think has been really important finding that we've made is that we're not only losing this reducible iron fraction, but we're also releasing organic carbon that was associated with it. So in these extractions, we're able to capture of DOC alongside the reducible iron. And there's a lot that goes into those extractions. So if people have questions about those, maybe they can ask those at the end. But what we find from doing those extractions is that in the PALSAs, in the deeper horizons, so we have a sort of transition zone and the very mineral-rich horizons, we have about 20% of all of the organic carbon is associated with reactive iron minerals. And when we go to the fen, we see that there's absolutely zero. So we have this mineral stabilization in our PALSAs that we just completely lose by the time we get towards the fen. And it's really interesting to look at this with nanosims. So these are the fine fraction which is separated from the soil in both the transition zone of the PALSA on the left and the fen transition zone on the right. And you can see in the PALSA, you have this kind of pink aggregate. And that means that we have a particle which is basically a co-precipitate of iron and carbon. So we've mapped the iron and the carbon on this very small scale. And if you actually kind of sputter down through this aggregate, you find that it's this iron and carbon the whole way through. It's not really like a coating. It's really like a sort of iron carbon mush. But when we move to the fen, we still have both iron and carbon, but they're more next to each other. They're not really associated. And we think that's coming more from the clay particles. And what's really interesting is that we see this loss of reactive iron in the solid phase reflected in the pore water data. So if you look at the iron 2 data and the DOC concentrations in the pore water, you can see that we have almost no iron 2 here in orange in the PALSAs, but we have up to 2 millimolar iron 2 in the fen, and that's very high. That's the highest I've ever measured in any environment that we've ever worked in. And this really high iron 2 concentration comes along with a very high peak in the DOC. And recently, Monique's been doing a lot of more recent work showing that you don't have to go all the way through the thaw transition for this to happen, that we actually start to lose some of this iron protected carbon into the aqueous phase very early on in the PALSA collapse. And maybe you're wondering when I was going to get back to talking about bacteria. We think that the main culprit for all of these observations is the activity of iron reducing bacteria. So we quantified the number of iron reducers that we find in all 3 horizons across the thaw gradient. And we find that between the PALSA and the fen, we have a significant increase in the abundance of iron reducing bacteria. And we specifically chose to do this with a cultivation-based approach rather than a molecular approach with something like 16S, because when you do a 16S survey, it's quite hard to pick out iron reducers specifically. So there's a few strains that are very well-known iron reducers, but there's often some microbes which are capable of doing iron reduction that we might not necessarily think as being classical iron reducers. And in fact, we did some isolation attempts from these fen soils, and we isolated one Geobacter species, which is a very classical iron reducer, so that oxidizes acetate. and does iron reduction. But we also isolated an iron-reducing cupriavidis species, which oxidizes lactate and does iron reduction through a kind of fermentation process. And if we picked this cupriavidis up in a 16S survey, we would never have said it was an iron reducer. So it just goes to show that sometimes doing a bit of cultivation can actually be really, really worthwhile, even though it's a lot of effort. So that's our story of what's happening in these permafrost peatlands. So we have a system in the Arctic where when you have raised palsas and intact permafrost, you can have quite a significant portion of your organic carbon that is tied up in these iron carbon kind of co-precipitates. But when we get thawing, we then get waterlogging, and we shift the entire redox regime into a reducing condition. We have microbial iron reduction and a lot of mineral dissolution that just changes the whole landscape of the aqueous geochemistry. So if we were to summarize how effective we think these iron oxides might be as stabilizing carbon, we'd say, okay, maybe they're quite good in intact palsa soils when you have intact permafrost, but it's only really effective in the early stages of thaw. So once we have waterlogging, then we kind of lose this sink and we rapidly get this mineral reduction. So I wanted to quickly give you an idea of what kind of things we're doing next, because we are really lucky to have a really fantastic collaboration on this project and a lot of really amazing students. So there's a bunch of students in Tübingen, so in Germany, where I was before, who are still working with Andreas Kapler, looking at, for example, the microbial processes on a molecular level that happen during the transitions between palsas and bogs. And Kita, who's looking at iron flocks and what their kind of carbon preservation potential is. We've even started to look a little bit at forests because there's actually a birch forest which is expanding onto our mire. And we're also expanding to other sites, which was some work from the master thesis of Hannah Joss, who's just finishing. We've been looking at Alaskan thaw lakes. I'm lucky enough to have a new contingent in Bristol who only started this week, so they are lucky that I haven't managed to take any pictures of them yet. They'll be on the next slide. But here we're looking at redox fluctuations and how that affects iron carbon interactions. And we've also started to move a little bit south and work in peatlands in South Wales. We also have some American friends and colleagues who are doing a lot of fancy mass spec to look at iron associated carbon and its fate. Also, we have 2 PhD studentships that are available to work in Tübingen. So if you think this is interesting and you think you might want to join our team and all these nice smiley faces, then you can ask me or send an email to Andreas Kapler, who is he'll be recruiting for his group. That's just all our collaborators and funding, who are all great. And I thought I'd leave you with some of the Northern Hemisphere wildlife.
Thank you very much, Casey, for your presentation. So Oscar, will you go?
Yeah, thank you very much, Casey. Now we will have, share the screen. It's the turn of Dr. Carolina Merino. Carolina is a researcher from the Laboratory of Conservation and Dynamics of Volcanic Soils at the Universidad de la Frontera in Chile, that has focused his work on microbes and carbon biogeochemical cycling. So please give her a warm welcome and let's enjoy her talk.
It's okay, my screen?
Yep.
Okay, yeah. First of all, say thank you for the invitation to present my work. The title of this presentation is Abiotic and Biotic Oxidation of Organic Matter Mediated by Iron and Manganese Reduction in Humid Temperate Rainforest. I work in the Laboratory of Conservation and Dynamic of Volcanic Soil, but we not only study volcanic soil, but as you can see, the volcanic soil corresponds to the majority of soil of southern of Chile. We have here 11 national parks under study, including the Antarctic. And today I go to show 5 areas that we have under study that included the Antarctic. And this classification that we saw here corresponds to the classification of FAO. For the introduction in Chile, most of the humid temperate rainforest stand in the Andes and coastal range and receive more than 5,000 millimeters of precipitation in the year, with a mean annual temperature of between 8 and 13. The climate factor, in combination with the organic matter chemistry and soil mineral characteristics, create an oxygen limitation that in general regulate the organic carbon turnover and ultimately sun accumulation. These forests are developed in soil with high manganese content that derive from parent material like metamorphic granite. And the interesting thing for us that in this forest soil that are under a strong annual precipitation, the anaerobic and abiotic effect on carbon and nitrogen cycle have never been studied. Now, in general, in contemporary anaerobic soil, nearly all of the reducing compounds are derived from soil organic matter and lithogenic elements, such as iron and manganese, that act like a cofactor for microbial metabolic activity. The origin of this soil property, as well waterlogged, are the main driver factors that contribute to the redox fluctuation that Casey spoke before. Here we saw that the fin pore regulate the diffusion of water and the soil organic carbon in the anaerobic microsite. And this anaerobic microsite represent here the hot pot where the abiotic and biotic zone oxidation occur. These anaerobic microsite are water filled and correspond to the fine pore site, retaining water at 15,000 hectopascal in general, and represent around 30% of the total volume of the pore space of the soil. Here we saw that iron and manganese are strongly influenced by the redox state and soil pH under anaerobic conditions that Casey mentioned before. With this information in this soil that we have under study, we work with data from some meteorological station and some sensor, like the redox sensor that we show here, so that you can get an idea of the large amount of precipitation that we have in this temperate rainforest. In this matrix of color, I don't know if the people see my mouse, but in this matrix color, the largest precipitation occur in June and July. In general, are about 450 millimeters per month. This is a lot. We seen in the precipitation of all the year are around 5,000 millimeters. And the other graph corresponds to the record of the redox sensor from the last month that we have in the field. This is the type of redox sensor that record data at 10 centimeter here, 25 centimeter, 40 centimeter, and 60 centimeter of deep in the soil. And with the record, we can show that the redox condition fluctuate a lot during the months from the moderate reduced condition that is here until the high reduced condition. For this, under the traditional assumption that the rainforest soil mineralized soil organic matter slowly due to the anaerobic microenvironment, particularly under high precipitation, because we we have the assumption that the more mineralization, for example, is in soil that have similar characteristics, but with less precipitation and high in temperature, like the tropical soil, for example, that have a lot of study in iron and manganese. We start to study the mineralization process in the temperate rainforest. Foresee how much is the release of CO2. We can show here in this graph that we study tree soil here, and we incubate soil in anaerobic condition, biotic and abiotic. When it's abiotic soil, we sterilize the soil, and when it's anaerobic, we maintain the soil free of oxygen. We can show that the mineralization under anaerobic condition is really high. Here, in the bar graph, show the percentage of this fluctuation under anaerobic condition. We also show here other greenhouse gases like methane and oxide nitrogen. Sorry, but all this is related at the idea that we have that biotic and abiotic mechanisms change really fast to maintain some balance in the forest between the accumulation and oxidation of soil organic matter when it's involved iron and manganese. Here there are a mean, for example, of the anaerobic mineralization that is 62% and the anaerobic is 38%. That is a lot. Working in the same area, we are also able to see how much microbial community change under oxygen limitation condition. And we do, for example, a PLFA analysis, and we can see how the content of total, how is the content of total negative, gram-negative and anaerobic bacteria increase under anaerobic condition, and how the fungal community decrease. This analysis have 6 hours of difference between the, we extract the oxygen in the incubation. It's really fast, the change. Also, we saw here other analysis that is the fluorescein diacetate. We can show that this is aerobic condition, and now in anaerobic condition, the activity does not decrease under anaerobic condition. The activity increase in this soil. Also, we evaluate in the first moment the enzymatic activity. And here also we saw an interesting change because the activity now change with a release of enzymes that are capable to decompose more recalcitrant organic matter like peroxidase release. With all this idea in mind, we began to work in the following research question. How do biotic and abiotic redox coupling of iron and manganese reaction under anaerobic conditions contributed to the soil mineralization process in humid temperate rainforest soil? Here we have the first study, what we do. This study is for the evaluate the mechanism of CO2 release from the soil in absence of oxygen, considering the Fenton reaction. This is the Fenton reaction. The Fenton chemistry is a reaction that involves the reaction of peroxide with iron 2 for carbon oxidation by the generation of this hydroxyl radical, which is a strong non-selective oxidant of organic matter compounds, particularly lignin-like structure. We study the Fenton reaction in combination with the activity of manganese, peroxidase enzyme, and lignin peroxidase for evaluate the contribution of this biotic and abiotic process to the soil mineralization under oxygen limitation. Here is the experimental setup. I will try to explain this experiment because it was a little crazy. I will show some photos also. It's a novel technique that we use when we combine level peroxide isotope in the oxygen, 18 oxygen and iron 2 to induce the Fenton reaction coupled to the enzyme activity. We induce the Fenton reaction in different rates of peroxide and iron, 1 to 1, 1 to 5, or 1 to 10. And we measure the leveled CO2 that is released, the iron solubilization, peroxide consumption during 6 hours in a closed circulation system that is this. We use 10 grams of sterilized soil for all as treatment with peroxide, iron, and in some case with enzyme or not. Sorry. This closed system trapped the CO2, and the air was transported here in a microcosm by a pump. The produced CO2 and the water pass through a tubing that placed in a container that is this, that is filled with dry ice and ethanol, and the temperature was maintained at -60 Celsius. In this container, the water is quickly frozen to minimize the exchange of this isotope into the water, because we want that the isotope is exchanged in the CO2, not in the water. And the CO2 is not retained here because the CO2 have a lower freezing point that is -68, and the CO2 continue for here. And we also put here a residual muster air flux for trap with a drying agent all the water that can remain from this process. And this the CO2 continue here, and here circulate is frozen in the closed system. This is a exotyner that is liquid nitrogen, and the liquid nitrogen have a temperature of -196 degrees. And in this moment, the CO2 is frozen here and maintained. We accumulate the CO2 in this system for 6 hours. And we can show here the system look a little crazy, but we can show the picture because all the system, all this flag, all the treatment are connected in the same system. Well, when we apply ethanol to the dry ice, look like Halloween because all is frozen. We maintain this for 6 hours in circulation. Here, some interesting result that we can mention of this experiment when we have treatment only with Fenton, with all the ration, without enzyme, and we have the same ration with different enzyme. Something important that I need to mention here is that we purged the enzyme from sigma aldrich. This is something that I will mention in the next experiment because it was an important point for us. No use enzyme that we purge it all the time. Here we can show that the exotyner that were immersed in dry ice and methanol collecting approximately the 1%, that is the red line here, of the CO2 produced. Doesn't mean that most of the CO2 produced from the soil was successfully separated from the water use the trap system, and the remaining CO2 was regulated in the CO2 trap. The CO2 released from the sterilized soil after 6 hours of incubation range between 39 and 659 milligrams of CO2 per gram of soil. And we can show here that the amended soil with peroxide and iron increased the total CO2 efflux in a 20% from the control soil of the application. But the more interesting here is, I have this. that adding manganese increase the CO2 only when the doses of phenton is not present. In the case of the lignin work very well with the doses of phenton. For what? Because for the lignin and for the manganese peroxide, the peroxide that is the substrate is the same substrate for the phenton reaction. And in this case, when we produce the phenton reaction in presence of manganese, the enzyme is inhibited. In the case of the lignin, work together. Here also we can show here is the CO2, but with labeled peroxide, that soil with the ratio 1:1, the CO2 derived from soil, and then with this isotope was over 7-fold higher without addition of enzyme. That with the lignin and manganese treatment, this was higher. And reduced oxidative capacity of manganese and lignin and their lower peroxide availability. However, the increase of the proportion here don't have a really tendency. We don't observe a systematic effect in the increase of the dosage of peroxide and iron, and with related with the enzymatic activity and the release of CO2. Here we can show that soil amended with lignin were closely linked to the efflux of CO2 and both consumed peroxide and solubilized iron. This relation was weaker for the soil with an enzyme and was almost absent and insignificant for the soil with manganese peroxide amended. Some conclusion that we can have in this experiment. Don't worry, I don't will explain all this scheme, but it's for you have an idea of the process of a chain of the labeled peroxide into the CO2. Because this figure that we have in the paper show the different steps of abiotic oxidation with the radical produced by the phenton reaction. In the first line, we see that the radical attack aromatic structure by adding one hydroxyl group here in the aromatic ring. In the second step, two subsequent hydroxylation reaction convert aromatic structure in the hydroxybenzol, that is this, that later change to a more organic common structure like maleic oxalic fumaric acid. And in this moment is produced the exchange of the radical with the organic matter and is produced two molecules of CO2. The conclusion that we can have from this experiment is that peroxide and iron mediate process that play an important role in the CO2 release and carbon destabilization in forest soil that anaerobic or fluctuant anaerobic condition can generate. In a second study that we do, we added two more national parks. In this case, like I mentioned before, we isolate the enzyme. We think that this is important for understand the biological process involved in the study. We isolate enzyme from white rot fungi that are here, that are part of the 5% of fungi that survive under anaerobic condition. Because if you remember the graphic of PLFA that I showed that in anaerobic condition, the population of fungi decrease a lot. Only a 5% survive. This white rot fungi is one of the species of fungi that survive. And we prefer to isolate them enzyme because the acclimatation, the stabilization, and the resistance that have this enzyme are different to the enzyme that we purchase. here also we we do maybe we love the conceptual model, but this is other conceptual model that we do for show the Fenton reaction and how this reaction coupled to extracellular enzyme. That this model show a cooperative mechanism between the zone oxidation by hydroxyl radical, hydrogen peroxide, like a electron acceptor for the both process for Fenton and for peroxide to iron 3 or iron 2. In addition, this radical convert to non-phenolic aromatic structure into phenolic aromatic structure that further react with enzyme to oxidize some organic matter. New format soil organic matter is a electron donor that are capable to reduce iron 3 to a ferric 4 during the Fenton reaction. In this study that we included more soil soil, we study the mineralization again with the Fenton and with the enzyme associated to the Fenton reaction, and we saw that the mineralization increased zip time with the peroxidase enzyme, and okay, and this was related also with the quantity of iron that the soil have. There was no effect on the peroxide ratio for the different doses of Fenton reaction. For this, we only consider one dose, and you can see that the metamorphic soil, that is the soil that have more iron, have the higher mineralization and have the higher consumption of peroxide. Also, in this study, we measured the reactive species with the hydroxyl radical. We visualize by laser microscope confocal using a proof for induced deflorescence. We can show here clearly that the control without the Fenton doses, that is this picture, showed the lowest fluorescent intensity that the treatment that have the dosage of Fenton. Also, we evaluate, we do a correlation with the relative fluorescence with hexanol for the presence of the radical, because the hexanol is useful proof for the presence of hydroxyl radical, because it's easily oxidized when there are this hydroxyl radical. Some conclusion that we can have of this second experiment was that in average, 31 of all the CO2 released by aerobic incubation was purely abiotic, and 69 was released through microbial respiration. That is a lot. Under anaerobic incubation, 70% was abiotic and 83% was related through microbial respiration. Manganese alindins coupled with Fenton reaction strongly increased some mineralization until long period of oxygen limitation in this forest. Finally, a third step that we need to explore in the forest was the bacteria, because like we saw in the first analysis, we saw that the gram-negative and anaerobic bacteria, and in general, in total, the bacteria increase when the soil is under anaerobic condition. And in this last study that I will show today, we included 2 more sites, plus to the site that we have before. And in this case, we included Torres del Paine National Park. That is a site that is in the southern corner, near to the something passed because my screen disappeared. I need to do something because there's a.
Just try to share again.
Yes, I don't know why. It all switch off. Wait. Ah, yeah.
There we go.
Do you see?
Yeah. Okay. Just put it on presentation mode, right?
Yeah. No? Okay. This is the last place that we include in the Torres del Paine National Park that is near to the Antarctic area. Is the final of Chile, really the final. And we included the Antarctic soil in the field Peninsula. Like before I told, we have a higher concentration of gram-negative bacteria and mainly of iron-reducing bacteria. We've seen that these iron-reducing bacteria play a key role for the lignin degradation in the soil under anaerobic condition. We want to assess the importance of this iron-reducing bacteria as a lignolytic degrader in soil with different parent material. For this, we included more soil with different, in a climate gradient and with different vegetation. Here we have this theoretical model that in general we use in the study for try to understand the representation of the concept. And in this theoretical model, the iron-reducing bacteria that work for lignin containing reduced in the soil organic matter, which is in turn served like an electron donor and like a carbon source for the same bacteria in a gradient of redox condition. Iron-reducing bacteria reduce insoluble iron 3 or oxydroxy to soluble iron 2. Also, this soil organic matter that is reduced can give an electron directly to the iron 3 as Fenton reaction, can oxidate hydroxide peroxide and produce during the aerobic phase to generate hydroxyl radical for soil organic carbon oxidation from soil organic matter. This is other way that we can represent the PFLA. This is as a screening of the anaerobic bacterial community by the PFLA. We also included here the Geobacter because we know that the PFLA is an analysis for the micro community, but there are some markets for some phospholipid membrane that we can use. Also, later we do obviously DNA sequencing for be sure that the presence of the bacteria because we doing a first screening of the community of the soil. Later we isolate this microorganism and we identify for doing the incubation in the soil with this bacteria. Again, this result was agreed with the similar order of the iron content in the soil and the CO2 and this generate the solubilization of iron in all the soil. The anaerobic mineralization that we have in this last study, we have a treatment that is a biotic treatment. Sorry, all is in sterilized condition all the time, o sea, in anaerobic condition all the time. This is a biotic system, a Fenton reaction treatment, and the treatment when we including the Geobacter. We can show here the total iron content. We can show that in the first national park of temperate rainforest soil, we have a lower content of iron, total iron, if we compare it with the Antarctic soil that have 57 grams per kilogram of soil of iron. The CO2 evolved from the sterilized soil system incubate that we added the iron reducer was 20 time higher than the other system, and the CO2 represents 60% of the total respiration of the no-sterilized soil. Like I told, the pattern was agree with the total iron content, iron 2, iron 3 in all the soil. And the lowest CO2 respiration that we have, we relate more this lower respiration with the higher aluminum saturation that is in the case of the granitic and metamorphic soil that represent the aluminum in the cation exchange site with the lowest pH. We evaluate the lignin degradation was analyzed by HPLC, which clearly showed that two no spike characteristics of the lignin chromatogram here. There was a good correlation between the control without the iron reducing bacteria and with the other soil that have the input of the bacteria. Again, the most degraded lignin treatment occur in the basaltic Antarctic soil that have the lower pH but have the high amount of iron content. Finally, we have a high and significant correlation between iron, total iron and CO2 was found in all the treatment. The slope of biotic soil was much deeper than for the abiotic sterilized one, and in average, the slope of the fluvioglacial and basaltic Antarctic was approximately 2 times more than in the other soil. Some conclusion that we can have from this study is that microbial respiration coupled with lignin decomposition under anaerobic condition was common in all the soil and was a maximal in soil with high iron content. Soil organic matter served not only as an electron shuttle and electron donor, but also as an organic carbon source. And the overall of climate and vegetation, respiration of different soil types show a high dependence of metal reducer microorganisms. Some perspective of all this work that I present, we think that anaerobic abiotic mechanism of soil oxidation play an important role, more than the normally is so. The combination between bacteria and fungal metabolite and abiotic and iron and manganese reduction or oxidation can generate reactive oxygen species, could contributing to lignin bedrock across of all the ecosystem. And we think that it's necessary to extend the study to other type of soil and other ecosystem in surface and deep rock. And I will tell thank you to my team. That's it all.
Thank you very much.
Thank you very much, Carolina. So a round of applause for both presenters. So now we have time for questions, and the idea, I want to invite everyone to use either the questions and answer button at the bottom of your screen is the Q&A. You can click on that and you can ask questions there, or you can also just raise your digital hand and we can allow you to talk and ask your questions directly. You can also use the chat, all of the options, and with Oscars, we're gonna be managing,
looking for questions,
organizing the questions.
Claudia has a question.
Yes, so
it has a real hand.
Thank you both for the great talks, and thank you Felipe and Oscar for organizing this the seminar series. I learned so much over the last months. My question was, you showed that the Fenton reaction produces organic acids, and
When I think about like temperate or boreal soils, there's a lot of organic acids in those soils, and I'm wondering how much, if you have an idea, how much of those acids come from vegetation directly, and how much comes from these weathering or biotic weathering reactions.
The truth is that we saw that this malic acid or citric acid was produced in the FAIR experiment only because we measure was only a circumstance that we measure this. We don't continue in this measure, and we never do with vegetation in the field. This is the step that we want to do now, do a Fenton reaction in the field. But it's really interesting how this can increase, but we don't have measure of this, only the present.
And I have another question for Casey. Do these ion reactions also explain the rapid loss of carbon from the British Isles? Because there was this big debate on, like, most of the British Isles are covered in peatlands, and there's been a huge loss of carbon, right?
I mean, I think there could be a small contribution for these iron-related processes there, but I think probably not. It's more land use change. So you have like a ton of, drainage and grazing and actual peat extraction. So I think when you have a natural system that's changing, then it can contribute to the carbon loss a lot, but a human activity will vastly outstrip that. Our peatlands are a mess, to be honest, but hopefully not for too much longer.
Francisco.
Okay, Cassie, thank you very much for your nice presentation. I have just a quick question. That the area that you showed there is a sediment or soil, because the parent materials, what is the parent material there?
It's actually lake sediment. So it would have originally been a lake bed that's drained, and then it's been accumulating peat for a few thousand years. So it's actually kind of half and half. So when you take these cores, our kind of mineralized horizon is lake sediment, and the organic layer is very much kind of peat. And then we have this zone in the middle where you have not so much a mixing of both, but you have a lot of iron that's released from the sediment, and it then becomes oxidized at these redox boundaries. So there's three very distinct horizons, and it's not really a soil. It's a sediment and a peat.
Yeah.
Technically.
And I would like to ask another quick question about why did you determine the iron by ditonite or oxalate? Also, did you use also oxalate? Huh?
No, we didn't do oxalate. So you probably know, there's 5 million of these extraction protocols, and trying to decide on which 5 to use is the total minefield. The dithionite extraction, there's a precedent of using that to determine iron-associated organic carbon. So what you do is you do your citrate dithionite extraction at neutral pH, which is important because it doesn't then precipitate organic matter at a low pH or carbon at a low pH. So you can measure the carbon that's released along with the iron that's released, and then you correct it for a control that's with sodium chloride with the same ionic strength. So the number that we get in the end of the iron associated carbon is the difference between what we measure in the extraction and what you measure in the control. But there's a lot of caveats about that. So I would say it's good to remember that it's operationally defined. So there probably is more organic carbon that's associated with iron, but actually getting a real number on that is kind of impossible. So we use a lot of different extractions to get a good idea what fraction is the most dominant. But there's problems with all of these extractions.
Yeah. Interesting.
Carolina and then Carlos.
What's related with the same that I saw, Cassie, that you show in the selective extraction iron sulfur iron S, and
Yeah.
I never saw this before. What is this? What type of extraction? What does it mean?
So that's not in our iron extraction, so we don't quantify that then, but we did it with a synchrotron method, so it's X-ray absorption spectroscopy.
Okay.
So that's an actual fit of what we think the minerals are, and we think there is some poorly crystalline iron sulfide, so it's not pyrite or something because that would show up, but we think we do have some sulfides in the system. That we actually haven't really investigated that much, but that's something kind of for the future. We weren't expecting that either, but we see it in a lot of samples actually.
It's pyrite.
Well, it's FES, so it's not crystalline pyrite.
So, Carlos.
Yeah, thanks. A very nice talk, both. Just wanted to ask first, Carolina, whether I would assume that these phantom reactions depend a lot on pH, and adding the peroxide obviously changes the pH. So what is the difference of pH of the soil among these different sites, and whether some of the differences that you observe have to do with the actual natural pH of the soil?
Yes, change a lot the pH. The Fenton reaction maintain the will of iron between 3 and 2, because when the Fenton reaction is produced, we saw, for example, in general, we have pH between 3.6 to 6 in the temperate rainforest. And we saw that when the Fenton reaction starts in the system, the pH can decrease around 1 point, and with this is produced more solubilization of iron 2 from the mineral, and they will continue because with more iron 2, the Fenton continue and continue. It is maintain the process of diffusion and saturation of water in this moment.
Okay, and for Casey, it's also about pH. Also, these processes had to do a lot with the pH of these organic pits there, right? What is the range of pH that you observe there in Sweden?
So in the bog regions, it's pretty consistently somewhere between 4 and 5. And then in the fen, it's around 7. So we see a lot of changes that are associated with this pH shift. So actually, I didn't talk about it much here, but what we actually see, especially in the fen, is we start to see a reprecipitation of iron, not in a sort of peat core or sediment core, but in like surface flocks in the water. So we think that there's these kind of chemical shifts that happen are also hugely changing the iron cycling.
Okay, so it seems to me like it's very important to look at the relationship between iron, redox potential, and pH. You know, it's like like these three things that seem to be tied together in this type of systems.
Yeah, they are completely tied together, and I mean, when you change the pH, it changes everything about the microbial community as well, because you have things that are just not going to grow at pH 4 that are suddenly really happy, which actually is probably why we see a big boost in the iron reducers there as well, because they tend to also, the ones that we are finding, they like the more neutral pH.
Okay.
Izzy, just a quick follow-up on that question. So why does the pH change so dramatically?
So in the fen, there's a sort of groundwater influx as well. And the groundwater is neutral pH. So it's a dilution effect, essentially.
Okay. So most of the water is coming from that groundwater source then? Yeah.
I mean, I don't know the flow rate through there, but it's not, it is moving through. So there's actually a lake, there's 2 lakes either side of the mire, and they're kind of connected. So you have a flow from one lake into the next one. And that's what's changing the pH quite dramatically.
Yeah, I was wondering because then you don't have a mass balance there, right? You have no idea how much is the input of the flow of this. No. Okay, so everything has been done in columns so far, so not really field measurement.
Yeah,
Okay.
so far in columns, and we've tried, because we've got enough cores now, we tried to make a sort of stock estimates, but that's really fraught with caveats because we have so much compaction with the cores. I mean, it's not so much compaction, we can estimate how much it is, but so you can make stock calculations, but I would just take them with a little bit of a pinch of salt.
Sure. Thanks. Claudia.
On the same note, so the redox is really tightly coupled to pH, because when you get into a different redox stage and you start oxidizing or reducing nitrogen, for example, that also affects the pH. So the redox reaction can increase the pH. Some of them are kind of neutral and some don't. So it's probably a combination of the water influx and changing the redox state. But this is super interesting.
Yeah, I mean, we find when we measure it, so the bog is really interesting in terms of the redox state, because most of the times when we've measured it in kind of snapshots, when we took these samples, it was really reducing. So you've got so much oxygen consumption, even this top layers are quite reduced. But we put some probes out, or a group that we're working with put some probes out last summer, and we saw that there was actually, because it was quite a dry summer, we then had a shift to an oxic regime in the bog, but the fen never changes. That's always really, really reducing. I think just to do with the water table height. So the fen, the water table is very much at the surface and it doesn't really fluctuate that much. Whereas in the bog, it's just under the surface and it's a bit more susceptible to those fluctuations.
For us, it's the same until now. The data that we have from different months only show that we have moderate reducing system, highly reduced, but the oxygen system is really not a lot. Maybe a little in the summer, but not a lot. In general, it's moderate reducing the system.
Cassie, one comment. It's interesting that you get about 20% of your carbon because of iron. Am I right? Or yes? Is the
Yeah, in the most concentrated layers, we see about 20%, which was actually a really interesting number for us because we're not the first people who's observed that. There was a similar sort of study with a Chinese group. So they did a transect along the Tibetan plateau, so kind of meadow sort of environment, kind of alpine steppe, but also a permafrost environment. And they also got about 20% of iron-associated carbon, and it's the same in marine sediments. So this number kind of, it comes up again and again. I'm not sure if it's just an accident that it's so consistent, but that's a maximum.
Yeah,
it's not as much as that in other places.
This is interesting because we also get about 20% in our volcanic soil. And the other 20% is aluminium. In total, we make an analysis about the metals can get or can retain more or less 40% of the total organic carbon. My question is, did you measure also aluminium in your system? Or?
We did measure aluminium. In the more organic horizons, the sort of transition zone, we don't measure a lot of aluminium, but the sediments have a lot of clays in them, so they do have aluminium in it. We haven't tried to quantify how much aluminium associated carbon there is, but that will make a difference in the more mineral soils. That said, we also don't get a lot of iron associated carbon in those soils. So I don't think it's a huge contributor, but we do definitely see iron, aluminium, organic carbon associations as well.
Okay, using pyrophosphate. Huh?
Yeah, we see some with pyrophosphate and with these dithionite extracts. But we were really surprised it was, so going back to this 20% number, we knew that was that much in kind of mineral soils, but then we were really surprised that you see it in organic soils as well, because I mean, it's still 20%. That means as a content, it's actually higher. And we're seeing that a lot in the soils we have from Alaska as well, that it's still sort of 20%. So the actual content of iron of iron associated organic carbon is actually higher in organic soils than it was in the mineral part of the same core, which is weird and quite cool.
Interesting. Thank you.
Do we have any more questions? No, in the chat. I have a question.
In the chat, there's one.
Okay.
So you say, or
okay, so, yeah, Liam had a question for Casey. Why the larger carbon stores are found in these organic rich permafrost peatland areas, do you think that the permafrost carbon in the continuous permafrost zone, more synergetic sites, shallower organic layer permafrost sites, where there's a greater influence from mineral material on organic matter, are more at risk of carbon loss following degradation due to this loss of protection of carbon from the iron?
So I think I got where that was going. That's something we've been trying to work on a little bit, is making, so where the work was at that point was, we have a correlation between a sort of loss of iron associated carbon, and in those same regions, I should say, you also have more carbon loss to the atmosphere. We haven't been able to determine yet how stable that sort of, we call it the rusty carbon sink. We don't really know how stable that is. So we see an association between iron and organic carbon, but we don't know if that's stable for a long time or if it's something that kind of forms and reforms. I definitely would say that there's a lot of evidence from the literature that releasing the carbon from this kind of mineral bound state would make it more available. And we have some first indications from the FT data, so the kind of high resolution mass spectrometry data, that the iron associated with carbon are things that you would typically consider quite microbially available. So we think that there is a sort of selection and a release of quite available carbon. So we think that I think the iron associated carbon, once it's released, is potentially more available than the carbon that was just there anyway, if that answers the question.
again, I will, if there is any more question, please raise the hands, write it. Even can be the question in Spanish. We can try to translate it and answer it. So there's more than one option to do the questions.
So I have my question. I can ask it now. So it's for Carolina. And then I have another one for Casey, but after we see if someone else wants to ask. So why is the aerobic, in the last slide you showed, Carolina, why is there anaerobic biotic mineralization more responsive to iron 2 compared to the other treatments? Because when you compare all the slopes of the different treatments, that's the one that just is like, it responds very rapidly to the increase on iron 2. So why is that? What do you think is that in that treatment and on the others?
Yeah, because for us, the treatment that is called anaerobic biotic is the treatment that we put in anaerobic condition. We extract the oxygen in a system, but we maintain all the community. That you can have iron reducing, but you can have methanogenic or sulfur reducing that work under anaerobic condition. And this is one treatment. And in the other treatment, we have soil without oxygen also, but in a sterile condition, and we apply the iron reducing bacteria. Of course, the potential of all the anaerobic community will be more than this species, but not in the Antarctic. In the Antarctic, we maintain all the community in anaerobic condition, and we put the iron reducing alone and mineralize more, depend. But the explanation for the three forests, the three first forests, is that we maintain all the community. I don't know if this answers your question.
Yeah, I'm just trying to make the link. It's just sometimes you get lost in all these reactions, but yeah, I think I got it. Yeah.
Yeah, it's foresee the potential that the bacteria have alone, because we saw that the iron reducing bacteria, in this case the Heobacter that we have near to the sick species of Heobacter isolate, work really well for the with the Fenton reaction and with other reaction, and we want some the potential that have for mineralizate this carbon alone and in community. Because in some moment you have, when you have a species only alone, you not see the antagonist, and there are other species that in the presence of iron reducer like Heobacter can reduce the population because are antagonists and use the same resource also.
Okay, thank you. Do we have any more questions? Any of our panelists?
Any raised hand? I'm trying to
Okay,
look.
so I have my other question for Casey now. So, so after you look at this experiment, I mean, this setup you have, which is, I assume, very common on the area you're working, right? That this combination of different environments. So, what is the, I think because we're looking, for example, Carolina was looking a lot of CO2 emissions and maybe another greenhouse gases, but I see that a very large loss of carbon in your system is DOC. So what is going to be the impact of that DOC moving away from the systems? And it's probably not something people are looking into a lot. So what is your take on that?
So on the one hand, so I presented DOC data because that's what we measured, but actually we're also losing a lot of CO2 and methane from the mire. So there's really quite high CO2 and methane emissions that happen, especially during the bog, and you have some kind of shifts from going from a kind of CO2 dominated regime to the methane dominated regime when it's really waterlogged. So it's not just DOC, so there is a lot of greenhouse gases as well. But the DOC aspect is really interesting because we haven't actually followed the DOC out of the mire and seen what it does. It would be really cool next time we go to have a look at the DOC in the lakes. Oh, my lights have gone off again. But interestingly, that's what we've started to look at in our UK peatlands. So there's a massive problem. Claudia sort of alluded to it before, that there's been a huge amount of peat loss in the UK over the past sort of few decades. And a lot of that loss is DOC. And actually one of the places we're working in is a reservoir that used to be a drinking water reservoir. And then because of the peat drainage, the DOC influx into the reservoir means that it's just not usable anymore. So there's about 50,000 people who had to get their drinking water from somewhere else. So what we're looking at here is about how the iron minerals might be able to sort of work in reverse and re-trap some of that carbon that's getting lost as DOC. But yeah, it's a super interesting question. It's really important. It's already having a big impact on a lot of aquatic systems.
That's pretty cool. I think we're going to see more and more iron and DOC building up in all these aquatic systems. And I think the impact could be quite large. I have family in Alaska, and the water quality has been, getting worse and worse with time, and it's mostly iron and DOC. so I think it's gonna become big, and so
everything is brown. All the water is brown now. It's awful.
So I don't know if we have any other more questions.
Not from the public. So
Great.
I will give the final words to the organizer of the webinar, and I'll also give a big applause for everyone, especially the panelists and the organizer, Felipe. Thank you very much for this month of new knowledge.
Oh, well, thank you very much, everyone that has joined us. We have a very, very diverse group of people from many countries that are joining us every week. So it's very exciting to see so many people, joining a webinar at a very, from different places and at very different times. Well, you guys some are finishing their days, we're starting our day. So I think it's pretty cool to see this community of excited scientists, and we really hope we keep in touch. I think there's a lot of cool research and ways for interaction between these different groups. Very cool questions to be answered, and very cool field trips to, you know, perform. So I think it's great to see everyone getting excited on this topic. So thank you, everyone, and we hope to see you around. And all the best. And thank to all our panelists that were great, just great. Thank you very much.
Thanks, Felipe.
Thank you, guys.
Thank you.
Bye, everyone.
Bye-bye.
Bye.
You. Bye, everyone.
Take care.
Thank you.
Bye-bye.
Bye-bye. Felipe.
Yeah,
might
you asked the question about the
record it.
videos. Yeah, we'll have to send an email. I haven't figured out